Hello, this is Arlene Perez, Grand Canyon University, Concepts of Biochemistry, and this is my presentation on glycolysis, gluconeogenesis, and the role of aldolase gene expression. Glucose is a 6-carbon molecule. It is a carbohydrate, which are molecules used by organisms as an energy source. Carbohydrates power aerobic and anaerobic cellular respiration into simple and complex molecules. Too much glucose in the body gets stored as glycogen, which is a polymer of glucose. Glucose can be made through glyconeogenesis, a process involved in the breakdown of proteins and fats. There are several ways to make glucose, which is very important in maintaining homeostasis. In the body, glucose travels through the blood to tissues needing energy. Then it goes through a sequence of biochemical reactions, releasing energy as adenosine triphosphate, ATP. When ATP is made, it fuels every energy requiring process in the body. Most energy comes from an aerobic or oxygen requiring process that begins with glucose. Glucose gets broken down through a process known as glycolysis, which is a non-oxygen or anaerobic process to make ATP and pyruvate. In this presentation, I will explain glycolysis, the two phases of glycolysis, NADH molecules and glycolysis, explain gluconeogenesis, gluconeogenesis pathways, aldolase involvement in both glycolysis and gluconeogenesis, the different isoforms of aldolase and their different expression patterns, and examine how aldolase B expression can be regulated by transcription factors HNF1, HNF2, C, EPB, and DBP. Glycolysis is a sequence of reactions that extract energy from glucose. It splits glucose into two 3-carbon molecules known as pyruvates. It is an ancient metabolic pathway that evolved many years ago and is done by most living organisms of today. Fermentation is a term used to describe the anaerobic breakdown of glucose or other organic nutrients to gain ATP energy. The first organisms came from an atmosphere that did not contain oxygen, so anaerobic breakdown of glucose was the method used to make energy. The primary step in cellular respiration is glucose. It is an anaerobic process done by both aerobic and anaerobic organisms. There are 10 steps in glycolysis. It occurs in the cytosol of the cell and has 2 phases, the energy requiring phase and the energy releasing phase. In the energy requiring phase of glycolysis, the beginning glucose molecule becomes rearranged and 2 phosphate groups become attached to it. A modified sugar, fructose 1,6-biphosphate, is made by this phosphate group. It is an unstable sugar, but it allows it to split in half to form 2 phosphate-bearing 3-carbon sugars. Since the phosphates used in this step come from ATP, 2 ATP molecules get used up. The 3-carbon sugars that are formed when the unstable sugar disintegrates are all different from each other. During this phase, only one glyceraldehyde 3-phosphate can either enter the next phase, but the unfavorable sugar can be easily changed into a favorable one, allowing them both to finish the pathway at the end. In the energy releasing phase, all of the 3-carbon sugars are converted into another 3-carbon molecule known as pyruvate in a sequence of reactions. During these reactions, 2 ATP molecules and an NADH molecule are made, since this is the phase that takes place twice. The first time forms each of the 2 3-carbon sugars, and the second when it makes 4 ATP and 2 NADH molecules. Its specific enzyme catalyzes all reactions in glycolysis. The main enzyme in regulating glycolysis is phosphofructokinase. This enzyme catalyzes the formation of the unstable 2-phosphate sugar molecule fructose 1,6-biphosphate. This enzyme speeds up or slows down glycolysis when the cell needs it to. Ultimately, glycolysis changes one 6-carbon glucose molecule into two 3-carbon pyruvate molecules. The total products are made from this process are 2 molecules of ATP. 4 total ATP molecules were produced, but 2 ATPs were used. There are also 2 molecules of NADH made. At the conclusion of glycolysis, 2 ATP, 2 NADH, and 2 pyruvate molecules are made. If there is oxygen available, pyruvate can be oxidized to carbon dioxide in cellular respiration, producing more ATP molecules. The NADH molecule gets cycled back and forth between NAD plus and oxidized NAD and NADH, which is a reduced form of NAD. Glycolysis needs NAD plus to accept electrons during a specific reaction. If no NAD plus are available, then this reaction cannot happen and glycolysis stops. Therefore, cells need to be able to turn NADH to NAD plus so that glycolysis can continue. There are ways for this to happen. The first is with the presence of oxygen. When oxygen is present, NADH is able to send its electrons through the electron transport chain, making NAD plus, so it can be used in glycolysis. During the electron transport chain, more ATP is also made. The second way to make NAD plus occurs when no oxygen is present. In this pathway, NADH gives its electrons to an acceptor molecule. This reaction does not make ATP, but does make NAD plus, allowing glycolysis to continue. This process is known as fermentation. Fermentation is the main metabolic strategy for many bacteria. There are even cells in our bodies, like red blood cells, that utilize fermentation to make their ATP. Gluconeogenesis is a group of metabolic reactions that occur in the cytoplasm and mitochondria. It maintains blood glucose levels while in a fasting state, similar to glycolysis, but the processes are reversed. Balancing, stimulating, and inhibiting hormones regulates the rate of gluconeogenesis. Glucose is the main source of energy for eukaryotes. If it is not available, organisms metabolize glucose from other non-carbohydrates. Gluconeogenesis converts pyruvate into glucose. There are 3 steps in gluconeogenesis. The first step for gluconeogenesis is to convert pyruvate into phosphoenolopyruvic acid, PEP. There are several enzymes involved in this process: PEP carboxykinase, pyruvate carboxylase, and malate dehydrogenase. Pyruvate carboxylase, found in the mitochondria, changes pyruvate into oxaloacetate. Malate dehydrogenase converts oxaloacetate into malate. This needs to be done since oxaloacetate cannot cross the mitochondrial membrane. After malate crosses the mitochondrial membrane, it goes to the cytoplasm where it converts back into oxaloacetate through another malate dehydrogenase. Finally, oxaloacetate is changed to PEP through PEP carboxykinase. Then the following steps are similar to glycolysis. The second step that is different from glycolysis is that it changes fructose 1,6-BP to fructose 6-P through the enzyme fructose 1,6-phosphatase. The same enzyme, phosphoglucoisomerase, used in glycolysis converts fructose 6-P to glucose 6-phosphate. The final step, where it differs from glycolysis, is that it changes glucose 6-P to glucose, utilizing the enzyme glucose 6-phosphate, located in the endoplasmic reticulum. Because it is important for organisms to conserve energy, if there is extra energy available, gluconeogenesis is inhibited. If energy is needed, then gluconeogenesis is activated. Aldolase is essential in the breakdown of triglycerols. It does this by stimulating the breakdown of glycerol. Aldolase changes glycerol into dihydroxyacetone phosphate, which allows it to enter the glycolytic pathways necessary for energy production. This production allows the body to use energy stored in triglycerides more efficiently. It is this that makes aldolase an essential enzyme in lipid metabolism. A critical function of aldolase is its aptness to reverse the function to maintain the homeostasis of glucose in the body. During glycolysis, it converts dihydroacetone phosphate and glyceraldehyde 3-phosphate into fructose 1,6-biphosphate. In gluconeogenesis, it catalyzes the disintegration of fructose 1,6-biphosphate back into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, allowing the body to efficiently make glucose out of a non-carbohydrate molecule. There are different isoforms of aldolase that are tissue-specific in their patterns of expression and cellular energy metabolism. These different aldolase include aldolase A, aldolase B, and aldolase C. These enzymes are vital for different metabolic needs and processes when regulating particular tissues. These isoforms have slightly different kinetic properties, allowing them to better process specific metabolism requirements for the different cells and tissues. This process can be seen in aldolase A. Aldolase A is critical during glycolysis and in energy production. They are located mainly in muscles and red blood tissue cells. During gluconeogenesis, aldolase B catabolizes fructose. Aldolase B found primarily in the liver and kidneys. In the regulation of cellular energy metabolism, the diversity of aldolase isoforms allow metabolism to occur through the various organs in the body. Mainly in the liver, aldolase B is highly regulated by a few transcription factors such as hepatocyte nuclear factor 1, HNF1, FOXA, HNF3, CCAAT enhancer binding protein, CEBP, and D binding protein, DBP. HNF1 is a transcription factor that is crucial in the regulation of liver-specific gene expression. This includes aldolase B. CEBP and DBP are another important factors that also help in the regulation of aldolase B expression. This is done mainly with their interaction with a particular promoter element found in the aldolase B gene. HNF3 also influences aldolase B expression. It is mostly through liver-specific gene regulation. It is not as influential in the regulation of aldolase B as HNF1, CEBP, and DBP are. With these combined transcription factors, they provide the particular liver-specific expression pattern seen in aldolase B. These transcription factors stick to a particular DNA sequence. Within the aldolase B promoter area, this determines the rate of transcription and eventually the amount of aldolase B protein made. So to conclude, glycolysis is a sequence of reactions that extract energy from glucose. It splits into two 3-carbon molecules, pyruvate, an ancient metabolic pathway It is a 10-step process that occurs in the cytosol of cells. There are two phases of glycolysis: the energy requiring phase and the energy releasing phase. In the energy requiring phase, glucose becomes rearranged. The two phosphate groups become attached to it, and unstable fructose 1,6 biphosphate becomes modified through a phosphate group. It lets sugar split in half, forming two phosphate-bearing 3-carbon sugars. It uses two ATP molecules. One, glyceraldehyde 3 phosphate, enters the next phase, changing to a favorable sugar, allowing both to finish the pathway at the end. In the energy releasing phase, 3-carbon sugars are converted into 3-carbon molecular pyruvate. Two ATP molecules and an NADH molecule are made. Specific enzymes catalyze all reactions in glycolysis. The main enzyme, phosphofructokinase, speeds up or slows down glycolysis when the cell needs it. Glycolysis changes one 6-carbon glucose molecule into two 3-carbon pyruvate molecules. The product made are two ATP molecules. 4 ATP molecules are produced, but only 2 ATP molecules are used. 2 NADH molecules are also made. NADH gets cycled back and forth between NADH plus and oxidized NAD and NADH. Glycolysis needs NAD plus to accept electrons during a specific reaction. If no NAD plus are available, then reactions cannot happen and glycolysis stops. Gluconeogenesis is a group of metabolic reactions that occur in the cytoplasm and mitochondria. It maintains blood glucose levels when in fasting state. Like glycolysis, but reversed, it balances stimulating and inhibiting hormones, regulating the rate of gluconeogenesis. Gluconeogenesis converts pyruvate into glucose. There are 3 steps in gluconeogenesis. These are essential for organisms to conserve energy. If extra energy is available, gluconeogenesis is inhibited, and vice versa. Aldolase is essential in the breakdown of triacetylglycerols. It changes glycerol into dihydroxyacetone phosphate so it can enter glycolytic pathways. Its main function is its aptness to reverse its function to maintain homeostasis of glucose. In glycolysis, it converts dihydroxyacetone phosphate and glyceraldehyde 3 phosphate into fructose 1,6 biphosphate. In gluconeogenesis, it catalyzes the disintegration of fructose 1,6 biphosphate back into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. The different isoforms of aldolase are vital for different metabolic needs and processes. They have kinetic properties, allowing for better processes of specific metabolic requirements. Aldolase A is critical in glycolysis, Aldolase B in gluconeogenesis. The diversity of aldolase isoforms allow metabolism to occur through various organs in the body. Aldolase B is mainly in the liver. It is highly regulated by a few transcription factors. HNF1 is a transcription factor that is critical in the regulation of liver-specific gene expression, including aldolase B, C, EBP, and DBP transcription factors. Also helps in regulation of aldolase B. mainly with an interaction with a particular promoter element found in Aldolase B gene. HNF3 also influences the Aldolase B expression, but not as influential in the Aldolase B. These factors stick to particular DNA sequences within the Aldolase B promoter area. This determines the rate of transcription and eventually the amount of Aldolase B protein that is made. I hope you enjoyed the presentation. The following slides are references to this presentation. Thank you.